Hello, and welcome to your first fully vodcasted lecture for the semester. We'll actually have 5 vodcasted lectures when I'm gone for varying ski team events, including our nationals competition. I do have a couple of announcements for you, and if you are watching this during the regularly scheduled lecture time, then it is indeed Friday the 13th. So we're looking ahead towards next week, and a quick reminder that if you do complete the pre-test/knowledge survey by Tuesday the 17th, you will earn a replacement score for your lowest homework at the end of this semester. Remember, this is just one of many ways that you'll have to earn some extra credit this semester, but it is a really cool opportunity to get a quick preview into what will be coming this semester, as well as an awesome opportunity to help out with some knowledge that we will gain about your confidence and allowing us to facilitate your learning. And of course, do recognize that these data will only be used as a conglomerate of amongst the entire class to enable us to look at overall averages and whatnot. That being said, another note is that on Tuesday you will also have your first homework due. I encourage you to go ahead and get started and get that homework rolling so that on Monday night, if you'd like to come to my office hours and ask me questions, I will have those, even though Monday is a holiday. On Tuesday night, of course, I will also have my office hours, and you'll want to make sure that you're all ready to go before you get into that homework submission tab and actually submit your homework answers. In terms of lab, due to the holiday, we will not have lab on Tuesday. The reason for that is that we have actually labs on both Mondays and Tuesdays, and it would kind of set us off kilter if we were to have lab on Tuesday but not on Monday. So that's the reason that you're going to have an extra day off. That we had talked about that a little bit in lab. So do have your first pre-lab ready for the Thursday of next week so that you can be ready to go and ready to rock on that first lab. So we will be doing lab procedure number 1 next Thursday. Without further ado, let's jump into our very exciting material for the day and talk a little bit about pathogenic microorganisms, that is to say, those microorganisms that cause disease. So I'm starting off with what my prior students have told me is a serious no-brainer of a question. Which one of the following killed the most Americans? A, World War I, B, World War II, C, the Korean War and the Vietnam War combined, or is it D, the flu between 1918 and 1919? So, yep, we're in micro, and it is D, the flu between 1918 and 1919. But what you may not have realized is that the flu in that single year killed more than A, B, and C combined. In fact, it's estimated that between 1918 and 1919, there were between 22 and 40 million people that died of the influenza virus. On American soil alone, there were actually more than half a million individuals who were killed by that disease. If you compare that to something around 116,000 that were killed in World War I, you start to get the feel that really the 1918-1919 influenza was incredibly deleterious. Okay, let's, on that note, talk about sort of the historical look of infectious disease. And we could say that over time, those disease-causing microorganisms, we call them pathogens, have been formidable enemies. If we look back even further in history, between 1346 and 1350, there were actually 25 million people that died of the bubonic plague. Now, you may not realize it, but that's caused by a bacterium called Yersinia pestis. And so that caused a lot of deaths. If we look at smallpox, over the last 4,000 years, we've seen the death of some 10 million individuals. So it's been quite a battle against infectious disease. And in fact, even if you were to go onto the CDC's website and check out the leading causes of death in 1906, you would see that in fact 3 of the 4 leading causes of death were infectious disease. Number 1 was tuberculosis, number 2 was pneumonia, and number 4 was diarrheal diseases. For example, cholera. A few years back, actually in 2006, I went bike touring up in England, and I was up in the northern area of England. I was just south of York at this time in a place called Selby, and I found this burial ground for cholera. So I thought this was a good example of the sort of epidemics that could occur from just diarrheal disease. And you see, this was a plaque put up in memory of those individuals of Selby that did die from cholera. And even though this was a small community, over 100 people died, and it was the trigger to inspire the waterworks that were needed to allow for clean water that didn't contain those diarrheal disease-causing pathogens. So this was, if after moving up north just a little bit, we came to York and we went to a museum there, and I thought it was interesting to see there then the number one cause of death in 1906, tuberculosis. But even more further, looking further back in history, this was the spine of a soldier that died of tuberculosis that had become systemic in the British Romano period. So even much earlier in history, that was quite a scourge. Okay, so yeah, 1906, infectious disease, pathogens were a huge and formidable foe. If we were to look at the leading causes of death on the CDC's website in 2000, we would find that none of the top three were actually infectious disease. So we could say that, many infectious diseases are said to have been quote unquote eradicated, meaning that they're gone. Now, whether we buy that or not, we can still say that, certainly those numbers have been decreased and we are no longer fighting infectious disease in the same way. However, I have a feeling that many of you who have interests in medicine are thinking, yeah, but isn't infectious disease still a major threat? Sure, it is. And I thought maybe Sandra has an interest in internal medicine, and maybe you're thinking about some of those newly emerging diseases that we still have to fight. And some of those that we think about are things like AIDS, still a hugely formidable foe. In fact, we would call it perhaps the pandemic of my generation's lifetime. SARS, West Nile virus, swine and avian influenzas, right? All of these could be put on a list of things that we're currently thinking about. Now, on another note, Jordan, he mentioned an interest in international travel, and so I thought this was a good time to bring this up. His passion is actually in Korea, and so I'm assuming you have also traveled there. And one thing that's interesting about all of our international travel is that it allows for the more rapid spread of disease. And here's a good example. I mean, in 2003, I was in China, and I spent a big portion of the summer in China. And when I left, I actually ate breakfast in Beijing. I flew then to Tokyo. I ate lunch in Tokyo. I ate dinner, a late dinner in San Francisco, and I was back in Laramie for a late night snack. So if you think about it, right around that time was when SARS was a huge problem, and in fact it was right after that it became newsworthy. And so if I had SARS on that day, I would have spread it to all of those places in one day. So because of international travel, also nonchalance with regards to vaccination, because of immunosuppressed older patients who are living longer, we are seeing a resurgence of many old diseases. For example, right here I have a newspaper article talking about the increase in Lyme disease, and that's something actually some of you may remember when President Bush got Lyme disease. This is something that is increasing. In fact, all vector-borne diseases, if you go on to the CDC's website, you can see again that vector-borne diseases are on the rise. So not just Lyme disease, but also malaria and dengue fever. And in fact, some of you may have been reading in the news about the Wolbachia bacterium that they're now finding has some ability to stop the spread of malaria. And so that was a fascinating, one of my student groups did a project, their poster project on that last semester. Now, lastly, I think is something that will probably interest Lauren, who has an interest in cancer research. Many chronic diseases are actually being shown to be caused by microorganisms or agents. And so, for example, it was in the 80s, about 1983, that people began to realize that peptic ulcers, we used to think, and in fact, maybe you've talked to your grandparents about ulcers at some point, and they say, oh, they're just caused by stress, right? Well, we now recognize that no, 90% or more of all ulcers are caused by a bacterium called Helicobacter pylori. Hang on just a minute, I have a stuffed microbe of Helicobacter pylori. Yep, here it is. This is my stuffed Helicobacter pylori. You can see last semester when I passed it around, some of my students decided to make it a little gangsta headband, and as well as a scarf, which I didn't know if that went with the gangster theme or not. But you can notice that Helicobacter pylori, the peptic ulcer causing bacterium, has flagella. And they also braided the flagella last semester, but the flagella are what we call lophotrichous, meaning that they come out of one end of the bacterium. And they give virulence to this bacterium because as those flagella rotate, they allow the bacterium to bore into your stomach lining and cause peptic ulcers. So that's one example of how bacteria are being shown to cause diseases that were not thought to have been caused by bacteria before. Now, the reason that I say Lauren may be interested in this is because it turns out that many types of cancer are also caused by viruses. So a good example of that is in the summer of 2006, and I bet many of you remember this. It was noted that human papillomavirus, or HPV, is the cause of a majority of cases of cervical cancer. Now, last but definitely not least, in fact, this is going to be a reoccurring theme all semester. One of the hugest things that we're fighting is antibiotic resistance. Because of overuse and misuse of antibiotics, we've seen a selective pressure placed on microorganisms, and so we're seeing a lot of antibiotic resistant strains. Many of you, in fact, may have heard of something called Mrsa, and I'm just going to jot that down. Mrsa stands for methicillin-resistant Staphylococcus aureus. And what this means is that not only is this form of Staphylococcus aureus resistant to penicillin, but it's also resistant to methicillin, which means that we have to go to much more powerful antibiotics, and often antibiotics that aren't very selective, meaning that they often make the host feel pretty lousy too. So this then allows us to begin to wrap up our first lecture, and I'm going to go ahead and talk just about a few broad things, because when we've hit on these sort of Hollywoodized pathogenic microorganisms, we tend to focus in on the disease-causing potential of bacteria. But in fact, most microorganisms are not disease-causing, and most of them are helpful to us. For example, those bacteria that cover our body surfaces are actually helping us to they're competitive with microorganisms that might cause disease, so they compete for space and nutrients and actually protect us in that way. Also, microorganisms are wonderful model organisms for study because, hey, let's face it, nothing reproduces faster. And so we can see literally generation upon generation upon generation, 10 to the 9th organisms overnight, and we can study the changes that can occur in those generations. So studying microorganisms is a very, very rapid and effective way to study certain pathways and whatnot. And what we usually find is that, in fact, there's a fantastic quote by a scientist named Jacques Monod, who says, what's true of an E. coli cell is also true of an elephant. And that's often very true because we see that pathways like glycolysis are fairly well conserved across the microbial world and into the world of eukaryotic organisms. Okay, that brings lecture 1 to a close. And as we dive now into the beginnings of lecture 2, a quick note. You'll notice that you don't have a full and complete set of notes for lecture 2, and that's on purpose because after I finish lecture 2, I will post the full set of notes. So I don't want you to rush to write down everything that I'm going to say. You can jot a few notes if you'd like, but on Monday, we'll be playing a game and we'll actually finish off this lecture. And so you'll be able to get the lecture notes when I post those. I'll even try to post the first few notes at the by the end of Friday so that you can get on and see some of the early ones as you're working on your homework.